Hello, my name is Kristen Chu, and I am the lab manager at the UCLA Brain and Body Lab. Today, I will be sharing a project titled The Role of Parent-Child Interaction in Emotional Processing, Alexithymia, and Mental Health. Alexithymia, a personality trait characterized by difficulty identifying and describing feelings, is associated with the impairment of emotion regulation and symptoms of internalizing psychopathology in children and adolescents. However, studies have found that more maternal care was associated with less difficulty describing feelings in children, and that greater use of positive emotions in parent child interactions was associated with less difficulty communicating emotions at college age. Additionally, parent socialization of emotion, for example, parent expressed positive affect, has been shown to play a significant role in improving youth emotion communication as well as protecting against youth internalizing psychopathology. Thus, parent child interactions may be important when considering child's emotional processing. It is unknown whether parent expressed positive affect in parent child interactions moderates the association between child's alexithymia and internalizing symptoms. Our three aims of this study were to 1. Determine if alexithymia symptoms are associated with internalizing symptoms in a community population of youth. 2. Identify whether parent expressed positive emotions in a behavioral measure of parent-child interactions are associated with lower youth alexithymia. And 3. Establish if parent expressed positive emotions in dyadic interactions moderate the association between child alexithymia and internalizing symptoms. For the methods, this study included a subset of children and adolescents aged 6 to 16 from data collected as part of a larger study. Data collection is ongoing, so these analyses should be considered preliminary. AIM 1 has a sample size of 83 children and adolescents, while AIM 2 and 3 have a sample size of 51 children and adolescents. We took some quantitative measures of alexithymia and internalizing symptoms through the Parent Proxy Reported Alexithymia Questionnaire and the Internalizing Symptoms subset of the Child Behavior Checklist. We also recorded videos of dyadic interactions where parents and children were asked to pick a pleasant event to plan out together. These videos were coded using the Validated Family Interaction Macro Coding System, also known as FIMS. Dyadic interactions were specifically coded for frequency and intensity of caregiver-expressed positive emotions. For our primary analysis, we ran multiple regression predicting child internalizing symptoms from child alexithymia scores controlling for child age and sex. We also ran a multiple regression predicting child alexithymia from parent-expressed positive emotion scores in dyadic interactions, also controlling for child age and sex. We conducted an exploratory moderation analysis examining the interaction between parent-expressed positive emotion scores in parent-child interactions and child alexithymia in predicting child internalizing symptoms. Moving towards the discussion, in this preliminary analysis, we saw that there was a trending but not significant positive association between child alexithymia and child internalizing symptoms. If you look at the top figure, the association between parent-expressed positive affect and alexithymia was negative, so more positive affect was associated with less alexithymia, which was the direction we expected, but the association is not significant in this preliminary analysis. However, when we did a median split of the alexithymia data to graph the moderation down in the graph 3 here, it is clear that there was less variability in parent-expressed positive affect, and there was also a lower average of parent-expressed positive affect in the high alexithymia relative to the low alexithymia group. So we hope to see whether that association persists or gets stronger when the data are analyzed as a full sample. Finally, we tested whether parent-expressed positive affect moderated the association between child alexithymia and internalizing symptoms, and this was not significant. So these data are promising, but associations are weak and we need the full sample size in order to properly test them. Once the full data set is coded, analysis will be rerun on the complete sample size and data-driven approaches will be explored as well. Specifically, rather than only look at parent-expressed positive affect, I plan to run an exploratory factor analysis on the dyadic data and look at how factors emerging from the dyadic interaction observational codes to see how these factors associate with alexithymia. This research will help to uncover factors associated with lower alexithymia and inform interventions aimed at improving mental health outcomes among at-risk children. Thank you so much.